Okay, perhaps we can get started on my little monologue about stroke and dementia. The examples I'll talk about are the stroke one has been published before. The dementia one is under review. The examples will give you an idea of how one can use the virtual brain in the context of looking at patients. It'll be a nice segue for the presentation on epilepsy, which will follow this one. So these are examples of strokes. This is not a good thing to have happen to your brain. So this is the same person up top here. You can see that the stroke is in the left parietal. You can see the T1 image where the tissue is actually necrotized. You've got additional atrophy and damage more anteriorly in the white matter. Down here is the middle cerebral artery territory, and you can't really see what there's actually an arrow here showing an inclusion in the artery where you get reduced blood flow. And this is obviously post-mortem of another person showing the tissue loss that accompanies stroke. Strokes are messy. They're not punctate lesions like you might get in neurosurgery, for example. So that means that the areas that are hit can be quite broad and heterogeneous. This is the distribution of the cortical vascular territory. Middle cerebral artery is the most common stroke that you tend to see. Anterior cerebral artery as well. These are coming from the carotid arteries. This is coming from the posterior artery, I forget what it's called, but basically coming up from the brainstem. When you see a stroke middle cerebral, you can get quite severe. I don't remember when this was done after the insult, but you can see the MCA, which is that lateral territory, knocks out a good chunk of brain. To model this in the virtual brain, you had an example this morning where you actually knock out an entire hemisphere just by knocking out the nose. And that would be an easy way to actually mimic a lesion, is just to take a bunch of nose and eliminate them completely. And that's actually probably the easiest way to model stroke. So for example, in modifying the Connectome, this is an example from an older version of virtual brain where you take out, this is basically the nodes in the MCA territory, and that's you just drop it out, and that really mimics essentially what you see when you knock out the areas in the middle cerebral artery. This is another depiction. You've seen this on various versions going forward. This is a quite extensive connectome. There's much more areas than we have in the previous versions. This is a control connectome, and then this is the non-control connectome. Do you see the difference? A or B? Which one do you want to be? Anyway. Okay. So that's probably the easiest way to actually model stroke, is just to get rid of the nodes. There are ways to do lesion mapping and incorporate that into virtual brain. It's currently not part of the workflow, but it will be as we start working towards modeling this. The difficulty, of course, with mapping the lesion is it's not really clean. So you've got a lot of tissue that's on the surrounding areas, the perilesional areas. It's not really clear how to model that because it could be healthy still, but it's really hard to know in the initial parts after the stroke. But you can get reasonable information from modeling stroke, and that's what I'll show you next. This is the workflow that was used for the particular model I'll talk about today. We have the structural imaging data. We've got the diffusion tensor imaging, tractography to generate the large-scale network. And then we have the T1 image here to generate the geometry. We also have fMRI bold data from these individuals. The models push through. This is the Stefanescu-Ursa 3D version. You have the K parameters, which correspond to different interactions between the excitatory and inhibitory pools of neurons. We have the local parameters and the global parameters, which are basically the scaling of the global coupling. Generates the local field potentials, which is pushed through the Bloom-Windkessel model, and then fit to the functional connectivity in this particular case for these individuals. So we had a question earlier about fitting to empirical data. This is an example of fitting to functional MRI. So the problem with stroke is there's chunks of tissue missing, which means that if you're going to do data processing to try and get regions of interest maps, for example, and generate functional connectivity, the conventional pipelines are going to have a problem because you've got a big hole where there used to be tissue. So the algorithms are going to have some problems dealing with that. So you heard earlier from Misha about the idea of doing a virtual brain transplant to help that. This too is not part of the virtual brain pipeline, but it is available, I think actually open source, to grab and apply to the stroke individuals. The paper itself that did this is from Anna Solokin. It's actually open source, so you can grab it if you want to see the details. And the notice here is that you've got a person who's had a stroke. You see the deformation in the brain area. The notion is to just basically remove that part of the brain and get a near image of the other hemisphere and then trace out where the lesion would be in that hemisphere. Now, it's obviously a coarse approximation, but at least allows you to run some of the pipelines that Misha talked about earlier today about being able to do the transformation into a atlas space to get regions of interest data and then derive the metrics for the individuals. This is just an example of trying to fit in the lesion into the tissue to make it homogeneous. You can go through the paper to get that. This basically, again, just allows you to be able to run the data through the conventional pipelines. You don't do much more than that other than to get do the tractography as well as being able to collect the functional data. It's not surprising there is a pretty dominant effect, a prominent effect of stroke on structural connectivity. And this is an example from a collection of patients from Inés Falcón, who used to work with Anna, looking at the change in the graph metrics for certain areas. Graph metrics now derived from structural connectivity, so use DTI to get the tractography and then do the graph metrics on there. A particular metric here is degree centrality, so how many areas are connecting to a certain node. And then a change in degree centrality across patients as a function of the extent of the lesion in precentral gyrus and putamen. I think these are all MCA territory lesions as well, which is very close to these areas. So you might expect there to be a change in their connections. So we do know that there will be changes in structural connectivity in areas that are away from the actual focus of the stroke. And that's not surprising because you will see degeneration in white matter tracts because they do get knocked out in stroke. It's also shown here. We've got patients and controls and looking for the change in the weights of the connections. This basically the weights go down, so you get a less connected network in strokes relative to controls. And this is a fairly large sample of individuals that they got from their stroke database. So again, in this particular instance, as I mentioned before, we're going to go back and use the Stefanescu-Yersa 3D model and model both the local changes in the parameter estimates for the excitatory and inhibitory interactions, as well as the global coupling. Conduction velocity as well as global coupling is going to be estimated in this particular case. So what's done here is sort of a two-stage process. First of all, you do a broad parameter sweep just to get an idea of how the model's working. That's something that Paul showed you earlier today, where you generate, actually everybody showed that, I guess running simulations, doing a parameter sweep of a large range of parameters just to look at the behaviors of the networks. Once you have an idea of sort of the areas in which the model's optimized in terms of a working point, where it has, for example, decent variance or good time series estimates, for example, you can then use the mapping then back to the empirical data and see if you can optimize it even more, fitting, for example, back to the functional connectivity, which I'll show you in a second. And these are kind of the maps you can get out of virtual brain to look at the the behavior of the system as a function of different parameters. And this is the global coupling parameters here. And this is conduction velocity up here. You don't really see that. And what this is showing is a control subject and one with a stroke. And the metric here is the total variance of the network, global variance of the system. And what you see happening here in the control subjects is as you move across a parameter space, you have a peak roughly here. So conduction velocity doesn't matter much, but coupling does in the control subject. The region in the stroke subject is much smaller and much more focal, suggesting that there's some dependencies have been changed as a consequence of the stroke. So you simulate the local time series and then push it through to generate functional connectivity. This is just basically this is the modeled modeled functional connectivity and the empirical functional connectivity. This is kind of messed up in terms of color scales. I apologize for that, but it's basically showing you that the pattern of functional connectivity is not too bad. The correlation here is non-zero at least. I think it's about 0.23 or something like that, this particular one. You can look at other aspects of the model. This is actually from the stroke individuals' data sets. For instance, you can look at the spectral power of the simulated versus the empirical data, amplitude, the overall variations in amplitude of the empirical and the empirical and simulated data, and then the actual functional connectivity itself. Again, the empirical data, simulated data, and the correlation. The maximum correlation we can get in this particular case is about 0.3 across the entire matrix. As you go up, the correlation comes to a peak, the fitting function, then it will actually drop off as you increase certain parameters. So there actually is a unique fit in this particular instance for the model. So again, you use the parameter sweep to get the rough idea of where you want to focus the model fitting, and then focus the model fitting on that and relate it back to some objective function, like, for example, functional connectivity. There are other things you can use, and we can give you some examples of that later. Just getting back again to the controls, we've got the global parameters again, coupling and conduction velocity. This is a different control that I showed you previously, a much narrower area in terms of coupling, in terms of the optimal fit. Here we have another control subject, the one I showed you before, and then 3 examples from stroke individuals. You see that the global properties are quite different in terms of the network dynamics that are enabled in the stroke individuals. In this particular study, there were 35 stroke individuals and a comparable amount of age-matched controls. If you look at the local parameters, roughly speaking, this corresponds to excitatory and inhibitory coupling. And this is a control again. And if you look at, again, the heat map, the maximum variance for the conjunction of the 2 parameters up in the corner there. If you look at our map of the controls on the top and the 3 patients, you see that parameter space is quite different in the patients. Again, not surprising, their brains have a massive hole in there, plus the structural connections are all messed up, so you're going to have changes in the dynamics that are enabled. The cool part about this was that the local parameters actually correlated with how well the patients responded to therapy, motor therapy in this particular case. This is a Fugl-Meyer index for motor rehab. And you see that the higher the excitation, the worse they did post-therapy. So higher excitation here, negative correlation means you're doing worse. This is immediately after therapy, and this is actually, I think, 1 year later. So it correlated both with the response immediately after therapy as well as the savings, if you will, of the response. And that's relating back to the excitation in these individuals. So it's kind of cool that in this very small sample you can get a notion that the parameters that you derive from TVB can be related back to something that's clinically relevant. In this particular case, the outcome of motor rehabilitation. This is being done now in a much larger sample, of course, because this is proof of principle. So you really want to validate this in a much larger sample and also look at the savings across a much longer period of time to see whether it's relevant. But again, I think this was just a nice sort of proof of principle that these parameters can be used for something beyond just the academic aspects of knowing this is kind of cool, I can model stroke, but now I can actually use the parameters directly to understand patient etiologies, for example, and potentially also use it for prediction. So, yeah.
So again, the question, I know there's no general answer, but how, in this case, did you make decisions on I'm sure that if you're not blinded, you could always generate such a result from when you have a lot of parameters to choose. So,
well, there were other ones that did not actually show the correlation, so they're specific. And part of it was driven by the fact that you do have both focal insult in stroke, but also, as I said, network effects. And the question was to come up with a model that can capture both of those things. The SJ3D is quite nice in the sense of it's being able to model a more biophysically interpretable aspect, which is this balance of excitation and inhibition. If you look at the change in the physiology of tissue that's in and around the stroke after the stroke does show this hyperexcitability as well, so it's nice to validate that in this particular application. So it's nice to show that connection as well. So.
And is it implemented a quantitative model fitting to find the best model?
Well, this one is a fairly. I don't think there's an actual fitting function per se in TVB yet.
No,
not yet.
Let's put in this case, for example, to get the best correlation between empirical and simulated.
Yeah. You basically run an exhaustive search and then just kind of go through and do the mapping of the empirical and simulated, and you think, okay, these ones are actually the ones that are at the peak. Part of the problem, of course, is that these are, even though they're complicated models, they're relatively simple in what could be done. You can imagine in a much more complex model where you've got more parameters to search, for example, like in the epileptor, that you might want to have a more guided search algorithm. And that's something that they're working on now in Marseille, is to use optimization toolkits to try and decide on the best approach to optimize model fit. It's not done now. Right now, sort of brute force. We've got a cluster. We've got HPC all over the place. Let's just throw 1,000 simulations and see what we get. So it's less intelligent, but it at least got us a reasonable answer. Reasonable answer. Maybe not the answer, but reasonable. Other questions before I move on? Okay. For dementia, this is very much a work in progress still. Dementia is a tricky one because of a couple of things. First of all, it's an incredibly heterogeneous sample of patients. Even just within Alzheimer's itself, it's quite heterogeneous. You can imagine if you're trying to look at all dementias, Parkinson's, Lewy body, FTD, and so on and so forth, that the way the disease progresses in an individual can be quite different. You may also have other comorbidities, like, for example, small strokes, hypertension, which also results in small strokes, a lot of white matter degeneration that happens sort of as a consequence of aging. I have white matter disease because I'm older than you, so I have these things in here that cause problems, and at some point there'll be other things that will happen as well, but for now I'm doing okay. The data I'm going to show you was from a paper that was just under review. We looked at a relatively large sample of individuals from an ongoing study from Australia, looking at healthy controls and 35 amnestic MCI and 16 Alzheimer's disease patients. There's a lot more that we're pushing through now, but this is sort of, again, proof of principle that we can get something reasonable from this. The model was using a very similar workflow that I showed you before with the stroke. We use a different model instead of this SJ3D. We use the reduced Wang-Wang model, which we heard about earlier today. And again, we're fitting to functional connectivity, optimizing the local and global parameters of the model. This is just another pretty picture showing you the workflow. Here we've got the diffusion tensor imaging to get the tractography measures, getting both streamline, basically connection strength, and distance, as well as, I can't remember which one's which. Anyway, generating the basic model itself, foundation model. You've got the Connectome, as well as the local dynamics from the Wang model. Generating simulated fMRI, getting the functional connectivity, and then fitting it back to the empirical FC, going back and forth until you've got, again, a peak to the fitting function. The key here was to also relate it back to the symptomology, the cognitive status of the individuals. So here we're showing the correlation between the model parameters and cognitive scores, which is basically a battery of neuropsychology tests that are used to assess cognitive function. I do want to show you one thing, though, that is important, and this is getting back to something that Misha was mentioning earlier today, is that these are hard data to work with because there's a lot of atrophy, there's a lot of misregistration between different modalities, and as a result, you can get ROI templates sampling CSF instead of sampling gray matter, which can be a problem, of course, if your ROI is basically sampling nothing, well, fluid instead of neuronal activity. So as critical as it is to check your data when you're doing healthy controls, it's even more so when you're dealing with patients, more so when you're dealing with older people who have large ventricles. This is an Alzheimer's patient, this is an MCI patient, this is a control. You can kind of see that the ventricles are getting larger here. I don't have an example of the bold data from this person, but there's a huge hole in their bold data from susceptibility artifact because of the space in the sinuses, and it blows out the entire frontal lobe, which if you don't check your data, you're not going to know that there's no data in the orbital frontal cortex for this subject, not because they have a lesion, but because the fMRI data is crap. Anyway, that's just worthwhile keeping in mind as you do this thing, because if the data you're using to model are a suspect, that means your model then by definition also is problematic. So be very, very careful about that. Having said that, this model turned out okay because we managed to get rid of the people who had severe artifact. And what we did is took a two-stage approach. We know in Alzheimer's disease that the first areas that are hit are in the limbic subnetwork, as you call it. Hippocampus, entorhinal cortex, perironal cortex, retrosplenial cortex are the nodes or subnetwork that first shows the manifestation of degeneration. So what was done was we first of all modeled the subnetwork first, a limbic network, if you will, and then did a second model on a full network and looked at the change in the parameter estimates as you go from a subnetwork to a larger network to see what the change was. So how much did the parameter optimization depend on how the model was embedded in the network? A model that's stable should show less of an effect of changing from a small scale to a large scale. If there's some problem with a particular set of nodes, it might change quite dramatically when you move it from a subnetwork to a broader network, which is what we found, in fact. Here is the example of the structural and functional connectivity for the full network and the subnetwork for the three groups. The subnetwork over here and the full network here. And you see primarily for the functional connectivity that does in fact go down on both the structure and function. You see in the structural connectivity that there is a change in the limbic subnetwork. You don't see that in the whole network level, which is interesting. If we now put that into an explicit test, this is using a multivariate analysis called partial least squares to do the mapping between the cognitive scores and the model parameters. The limbic subnetworks here, so you see up the top, those are the three local parameters here. Sorry, let me get the thing. The local parameters here, and then the global coupling here, and these are the 6 neuropsych measures. These are correlations with the confidence intervals. These are essentially Z-scores. And down here is the plot, scatter plot between the cognitive scores and the composite of the biophysical parameters. So again, limbic subnetwork, whole network, and then the difference, which is the embedded network I'm not going to go through the details of this, but basically the embeddedness, the change in the parameters was much stronger in the patients than in the control group. And if you looked at the variance of the parameter estimates within group, you see that the MCI shows the highest group variance, which you might expect because there's going to be some combination of those who are probably going to transition to show full-blown dementia and some who are not. And that heterogeneity shows up quite nicely in the parameter estimates, and you see that in the distribution of the parameter estimates within the groups. That might be actually the heuristic that can be used then to see who's going to transition versus who's not going to transition. The last slide was testing to see, is this really making any difference? Is it helping us to use this model or not? Or can we get the same information from just using structural connectivity or functional connectivity? So what this graph shows you is it's a resampling technique called Bootstrap. And this is looking at which of the models, or which of the metrics that we used has the best relationship between the cognitive scales that are being measured. This is measured based on the covariance, multivariate level covariance. So you can do the covariance with the structural connectivity, or SC. You can do the covariance of the functional connectivity, FC, with cognition, and then do the model with cognition. And do bootstrapping, which is basically resampling the whole full sample with replacement. So it tells you sort of how robust the estimate is, but also you can then compare distributions to see whether the confidence intervals around the parameter estimates overlap. So I'm showing you the data, the whole thing here. The distribution definitely is wider if you look at the model with cognition here, but you notice that the mean parameter for that is quite different from what you see in the distribution for the structural connectivity and functional connectivity. In fact, if you compare the confidence intervals and actually do a formal test on that, the model itself does much better in correlating with cognitive scores than does either functional connectivity or structural connectivity, which is kind of cool because it does suggest, first of all, that there is something useful in doing this modeling. It's somewhat paradoxical because you have to keep into consideration that what we're using to get the model parameters is, in fact, the structural and functional connectivity. So it seems kind of odd that you would get a better model by using the model than by using the empirical measures themselves. And I don't have a good answer to why that might be the case, but our suspicion is that you're actually extracting the useful variance from the empirical data by pushing it through the virtual brain model. And that confluence or conjunction of the biophysical parameters that are representing the local dynamics plus the long-range connections that are coming from the structural connectivity, it's extracting the most important signals from that to construct your model. You don't get that in the structural connectivity by itself. You don't get that with the functional connectivity by itself, but you do get that by using the model to eliminate essentially the erroneous variance. And it's likely the case here. That's why you get a better prediction with the model parameters than with the empirical measures by themselves. At least that's my best guess so far. So with that, I will pass on the baton back to Paul and let you get your hands dirty by playing with some dementia and stroke stuff. Thank you. Is there questions while we're doing the transition? No clap, no clap. Wasn't that good? Yeah.
I was wondering, since you're kind of looking at diseases that have this kind of important vascular component, are there any times to maybe incorporate data about blood flow into the workflow or into the model?
Probably into the workflow first, not necessarily in the model, just because it becomes complicated to figure out how to actually account for the heterogeneity of the vasculature. You can do perfusion imaging to get an idea of the vascular reactivity, and we do that now routinely in a lot of the Alzheimer's and aging studies, just to get an idea of is there vascular problems as well. But currently we're not using that in the model, but you could for sure. Go.
General strategy, on which level do you do the group comparison? And here you always generate the parameters from the model and from other techniques,
Mm-hmm.
and then you compare them on that level. But does it
I think the notion there is to try and, if you can avoid categorization and treating them as if they're a homogeneous category, it's probably better. We did, like, so in the, come on, oops. So even though you see the groupings here in terms of the colored dots, it was actually not treated as three separate groups. It was treated as a continuous variable. And that allows you to get a bit more of an idea of when you do treat them as one large population, do they naturally stratify that way, or are there some overlap? And you see that quite nicely with the MCI, is that they do, in fact, there's some that are clustering more with the Alzheimer, some that are clustering more with the controls, which you might expect. If we tried to treat it as three different categories, we probably would miss some of that. So I think it's important to try and avoid that if possible, because especially if you have clinical populations where there's heterogeneity like this. Three, two, one. Okay, go.
Did you try to apply machine learning techniques to try to classify these populations based on the model?
No. This is primarily because this is...
This is a clinical application, potentially.
Indeed. So what we're hoping to do is now there's actually a larger project ongoing that's using a much larger data set. So including the Sydney data set, we're using the ADNI data set, we're trying to get some additional data set in Germany to try and apply this in a much broader sample to see what are the features that are actually predictive of conversion to cognitive impairment. And that's where being able to model, to combine this with machine learning would be fantastic. But with this right now, we're just kind of seeing if this is going to work, and then for sure, we'll move on to the machine learning things. We're done. I'm done. Okay, Paul, thank you.